Hello everyone and welcome back to another video. In today's episode, we're going to be discussing a biochemistry, cell biology topic, levels of protein structure. Proteins, from a chemical perspective, are by far the most structurally complex and functionally sophisticated molecules known to man. In this video, we're going to be discussing the basic structures of these proteins. So without further ado, let's get right into it. So quickly, before we get into the levels of protein structure, I want to briefly discuss what a peptide bond is. And that is the covalent bond in between two adjacent amino acid residues. So in essence, a protein molecule is made from a long chain of these amino acids held together by covalent peptide bonds, which are formed through dehydration synthesis, meaning the loss of a water molecule. So therefore, proteins are referred to as polypeptides or polypeptide chains. So for example, one molecule of human insulin is going to have the same amino acid sequence as every other molecule of insulin. So let's start with primary structure. Primary structure of a protein is the precisely defined amino acid sequence that makes up that protein. So you can think of the primary structure as the individual amino acids linked together by peptide bonds. A covalent peptide bond forms when the carbon atom of the carboxyl group of one amino acid shares electrons with the nitrogen atom from the amino group of a second amino acid. What we see here is primary structure, and we'll go through each of these: primary to secondary to tertiary to quaternary. And we're going to go through each of these individually. Like I said, primary structure, the individual amino acids that make up the protein. When you break it down to the structure, you refer to each individual amino acid as a residue. So for instance here, this is tyrosine because of that R group. Second residue is glycine. We know this because it's the only amino acid that doesn't have a chiral alpha carbon. See the two hydrogens right here, makes us non-chiral or achiral. And lastly, alanine, the second most simple amino acid. So here's another example of breaking down primary structure into individual amino acids. This is something you'll have to do in biochemistry. So here's the first residue with the peptide bond. I'll draw right there. So that's the first individual residue. This is going to be alanine. Second residue, splitting it at the peptide bonds. This is proline. The third residue, again splitting it at the peptide bond. This is histidine. And lastly, the fourth residue is asparagine. So secondary structure is the localized folding into alpha helices, beta pleated sheets, along with beta turns and omega loops. And secondary structure is mainly held together by hydrogen bonding, which we see here, hydrogen bonds, depicted by the green dots. So you're going to see secondary structure mainly depicted by ribbon diagrams. Right here we have the alpha helice, and these are beta pleated sheets. These would be parallel because they're going in the same direction, which we'll see in a moment. So beginning with alpha helices, they're stabilized by hydrogen bonds. So with the alpha helices, we see that the R groups are on the outside of the helix, and alpha helices are right-handed. The screw sense refers to obviously the direction with which the helix is turning. It can either be right-handed or left-handed. Alpha helices tend to be right-handed. The hydrogen bonds that stabilize these alpha helices are 4 residues apart, and the corkscrew nature of the helix is about 3.6 residues per turn. And here's a simple representation of the beta pleated sheets, a schematic representation. Beta pleated sheets can either be anti-parallel as exhibited here, or they can be parallel as exhibited here. Next we have tertiary structure. Tertiary structure is the regional 3D folding of the polypeptide into its complex shape. this shape is devoid of symmetry. Side chains are held together via non-covalent interactions, and with the exception of disulfide bridges, which are between two sulfur atoms on cysteine residues. So when we say non-covalent interactions, we're referring to ionic interactions, hydrogen bonding, and other weaker non-covalent interactions such as LDFs, London dispersion forces. With tertiary structure, we see globular polypeptides and fibrous polypeptides. These are the different interactions and bonds that we'll see with tertiary structure. Disulfide bridges, ionic interactions, LDFs, hydrophobic forces, and hydrogen bonding. When you put it all together, this is pretty much what the tertiary structure of a polypeptide looks like. Most proteins actually stay as tertiary structure complexes, while others can go on to that fourth structure, the quaternary structure. Quaternary structure is simply when multiple polypeptide chains come together to form an oligomer, which refers to just multiple polypeptide chains. And these subunits are held together by non-covalent interactions. Right here we see an oligomer. This is actually a tetramer, meaning there's 4 subunits, 1, 2, 3, and 4. So I hope this video summarized levels of protein structures for you a little bit better. Keep in mind when we're talking about proteins, they are the most functionally sophisticated molecules known. Proteins are anything from enzymes to structural proteins, transport proteins, motor proteins, storage proteins, signal proteins, receptor proteins, transcription regulators, and other special purpose proteins. So keep this in mind, it's a very broad field of science. And it definitely takes some time to study and work through problems. You'll be on a better foot if you just start learning the levels of protein structure and the interactions at each level. I hope this helped. We'll see you in the next video. Thank you for watching.